That's the other problem they have. So they spend a lot of money that way. Yeah, they're not really weeds. In fact, living in this part of the world, unfortunately, your seaweed flora is extremely boring. If you go to parts of Western Australia and Southern Australia, there are seaweeds there that are so intricately beautiful, you're just spellbound by it. I mean, it really is something to see. So, yeah.
At what point does a barcode sequence variation indicate a different species? You can have different populations of the same species isolated, and which would show a different barcode sequence. But at what point do you decide that those two populations are different species based on the sequence? It's cytochrome, I guess.
That's one of the barcodes. Cytochrome C oxidase subunit 1.
And that's one you use for algae? Or?
Typically for the red and the brown algae, we'll use CoA1, but we'll also use the ITS. So typically we use multiple markers. For the quick screening, we use CoA1 typically. Okay? And this gets into the issue of is there a threshold or not, and so on and so forth. And we don't tend to look at it that way anymore. We use analyses that cluster things. And we look at within versus between group clustering. And typically what you do find is that within a good species, the values are well below 1%. And between species are typically more than 4%. And so there's typically a large window. When you get into the small window, like the Latissimus I was talking about at the end, in the Pacific versus the Atlantic, that's where it starts to get more interesting. Because there you could be dealing with incipient species or speciation. And that's where we start going in and doing a lot more than just a DNA marker. We start looking at nuclear markers and chloroplast markers, and so it becomes more of a holistic answer to the question. It's not 1% magic. Those days are gone. Yeah.
What areas of like commercial businesses are interested in you, or what do you share with them? What do they look for results out of you?
I think the bioprospecting people are the most interested.
Yeah. Because
they want to know how many species are out there and where they're distributed and those sorts of things. Most other industries, I think we come back to the seaweed issue. And so I'll get calls from someone, oh, there's an alga on my fish and it's killing everything, and can you tell me what it is? And so we do a lot of that kind of stuff, just helping people out. But the industry that's interested in biodiversity specifically is the bioprospecting industry. And it's a huge industry. Not so much other industries. A little bit the food industry. So Acadian sea plants, for example, is interested in different types of Calophyllus they might be able to market and things like that. And so they have interest in that regard. Yeah. Mostly bioprospecting.
What in the Bay of Fundy are the bioprospectors interested in?
I think the Bay of Fundy, unfortunately, was bioprospected. There was a huge group at the National Research Council in Halifax when I was there as a master's student in the mid-80s. That's when it was, yeah. And they did a lot of bioprospecting of local things. That's basically what they were doing. I think part of the problem is that the thing I brought up where a lot of things might have been thrown out with the bathwater. And so it might be worth going back and looking at some of the things where they said, oh, that species had an interesting property, and now we know it's 3 species. Maybe someone should go back and look to see if one of those species is the one that's interesting. But most of the bioprospecting that's going on now is being done in countries that are developing countries, where the bioprospecting has not been put through the ringer quite as much as it has been here in Canada.
So when they're out harvesting in the bay, collecting in the bay, how can they differentiate or not collect everything? It's sort of like dragging the ocean floor, really, in many ways. You see them collecting seaweed.
For the seaweed, they're raking the ascophyllum. So it's a particular implement that cuts the ascophyllum up and brings it into the cloud. And so typically they're bringing up only ascophyllum and the seaweeds that grow on the ascophyllum. And so it's a fairly good harvest that way. It's not like some forms of harvest. Off PEI, when they get the chondrus, which is the Irish moss, that's a thickener that you use in your foods and things, they do drag great big rikes along the bottom of the ocean, and that can create a bit of havoc. And obviously you get a bit of conflict there between the lobster fishermen and the people who drag these big rikes along the bottom. Yeah.
So is the harvesting that goes on the Fundy sustainable?
I can't answer that question because I'm not an ecologist. I know there are ecologists that are looking at that question. I can tell you there's only one species of ascophyllum. And that's unfortunate. It would have been nice if there was something else. Yeah. So that's a question for the ecologists. In Terry Chopin at UNBSJ would know a fair bit about that if you wanted to email someone and get some input.
I was just wondering, you were saying that how there's many species in the area that can be confused with one another. So does the misidentification in this region, are there any poisonous species that you should be wary of?
There are two questions. Are there any poisonous seaweeds and are there any magic seaweeds? So far, no. Seaweeds don't seem to produce the types of compounds that have an impact on mammals. They do produce a lot of nasty things for invertebrates. They're the typical things that try to eat them. So
basically, if I go pick anything up off the shore and cook it, it'll be fine to eat?
it might taste like, some of it really is disgusting. There's a lot of brominated compounds in some of them. There's a lot of phenolics. But there's nothing known to be poisonous on the coast here from a seaweed perspective. That I can tell you. And a lot of it is very edible. And some of it...
Eat it raw. That,
oh,
because I know that's quite common.
Yeah, we chew on it all the time.
Yeah.
Okay.
So you're okay.
I think North America has a huge wake-up call. When you go to Japan, I've been there collecting, there's a museum for kelp. It's just kelp, nothing else. It's that important to their culture. They eat it every day with every meal. And we just haven't figured it out yet. That there's this huge healthy food source out there at our fingertips, basically. So, yeah.
What compounds have been isolated from algae that are used in human medicine right now?
That I don't know. Yeah, I can't help.
There are some.
There definitely are. There are a number of antiviral and antibacterial compounds that have been found, but it's not my area.
So these prospectors have been successful.
Yeah, mostly with
Some made
seaweeds.
some money, maybe.
The big thing from seaweeds is food, and then the gelling products. The carrageenans, the agars, the alginates, the thickeners, the emulsifiers, those are the big ones. That's where all the money is.
But not in medicine.
Not so much medicine. There is the phycoerythrin that's harvested as a dye, that's used as a medical dye and things like that. But,
Who do you think should be funding your research?
I just need a sugar daddy.
I mean, like, who would benefit, there should be some way to say this is why it's a beneficial pursuit.
I think, people are willing to fund what's hot. And so they'll fund the molecular toys and the molecular tools and the generation of molecular data. But they won't pay someone to actually sit down and tell you what species that molecular data is coming from. And I think that's a serious problem. Because if no one on the planet, we will get to the point where no one on the planet can identify anything. I think that is an unfortunate reality of where we are going. There are maybe 3 seaweed taxonomists in Canada. I'm one of them. There's a gal at Waterloo who works on seaweed, believe it or not. And there's a gal at UBC who's not even a faculty member. She's there as an adjunct. She doesn't even have a job. That's how few people can identify seaweed in this country. And is it important? Yeah, it is when a guy who is, I'll tell you the aquaculture story. They called me up, our fish are dying, there's algae all over the cages, and it's this acid algae, and we think it's the acid killing the fish. I said, well, I highly doubt it because that thing doesn't leak acid. It keeps the acid inside. This thing, you don't want to eat this one, by the way. It's got vacuoles with basically sulfuric acid and sulfur. And nothing eats it. Even your inverts don't touch it. And I said, I highly doubt that's your problem. Send it down and I'll take a look. Now, these guys are going to start throwing lime in the water or something, I don't know, to counteract the acid. They sent it to me and it wasn't the acid. They didn't even have the right kingdom. The acid alga is a brown alga, and the one that was growing on their cages was a red alga, Polysiphonia. And I said, no, it's just a Polysiphonia. It doesn't release acid. Well, why is it killing our fish? I said, well, they're probably suffocating. I said, they're probably blocking the flow of water through your cage, and, well, that doesn't make sense. And I said, well, do you have an oxygen meter? Yeah. Well, what are your readings at night? Well, we don't have it on at night. I said, well, algae don't photosynthesize at night and make oxygen. I said, at night they use oxygen. I said, if they're using the oxygen and they're blocking the flow of water through your cages, your fish are going to suffocate. I said, do me a favor and run your oxygen meter for a night. I said, when are your fish dead? Well, when we show up in the morning. So yes, taxonomy is important. You need people who can let you know what those things are. And I think It's kind of a shame where it's gone in this country. There have been papers published in every major nation on the importance of taxonomy and the lack of taxonomic training, the lack of taxonomic expertise, the lack of taxonomic funding, and guess what's happened? We just had one come out 6 months ago from the Canadian Council of Academics. And guess what's happened globally as a result of all those reports?
What's the timeline for collecting species to identification?
You could technically do it in about 2 hours if you were serious. They are actually developing technologies that will be much faster than that. I've actually seen, and this used to be the myth, this was going to be you'd go out in the field with your little thing and you'd stick the little piece of seaweed in the top and it would do it all. That was the dream. But we actually have machines this big now that will do the PCR reactions. And this big that will do the sequencing. And slightly bigger that will do the DNA extraction. And so we're getting close to a point where you're going to have a machine that won't be much bigger than this, that's going to be able to run a sample in 15 to 20 minutes. And so when you're talking about border crossings and things like that, this is going to become a reality. They're going to be able to check things very, very quickly while you sit in your car and... But we're not quite there yet, but it is happening. And the way that technology gets developed is a company sees they're going to have a market. So until the market is there, companies won't develop the technology. The market can't be there until the database is there. The database can't be there until the taxonomists identify the collections. And around it goes. But there's been a huge breakthrough because in the US, the USDA, US, whatever it is, Food and Drug Administration, whatever they are, FDA, something like that, they have decided that they are going to be using barcoding as a means of dealing with food issues, and they're going a step further. Every species in every food item has to be listed on the package. And you need to be able to verify what's in what you're selling. And this is going to be the way that that's going to happen, this kind of technology.
When you said there were species that are joining in the Arctic, one from the Atlantic and one from the Pacific, they're hybriding? They're
Hybridizing?
They're
interbreeding.
Yeah. And this is the first incident that you're aware of?
This is the first case where we've got very clear evidence of it in our seaweed world. We have some other little hints of it in some other seaweed, that it might be happening. Yeah. So it's not surprising that Atlantic species are going to the Arctic. Like I said, that's where we thought they all came from. The surprising thing is we're starting to see a huge Pacific influence. And I think that's going to be a real eye-opener.
Okay, maybe one last question from me. I was particularly interested in the invasive species you were talking about. Do you see any of those that are coming in right now, particularly the one that you mentioned spreading north of the pole now? What kind of effect would that have? It would displace other species? Do you have any idea how that would work?
The problem with introduced species, you don't know what they're going to do to an area until they get there. But you're definitely dealing with species being displaced. There's no question about that. I think the bigger issues are what they're going to do to food webs and food chain. What things are being eaten now versus what's going to be eaten then, and what things may or may not eat it. Yeah.
There'll be some effect, but we don't know what it is.
It's just a huge cascade. And the system, and this is the problem, humans, we overrate everything we do. The system will sort it out, and there'll be some new equilibrium, whether we want it or not, and whether we like it or not, and Mother Nature is going to try to run what happened. But whatever that new equilibrium is. So, yeah. Thank you very much. I enjoyed talking to you.
Now don't just run away. Remember, we do actually have refreshments outside here. Spend a bit of time looking around, and I hope you enjoyed it. Thank you.